right, let me make sure this is actually recording. Yep. So I'll talk over this for a little bit. This survey should still be open, so go ahead and fill out the pre-lecture survey. And it asks you a bunch of, I expect technical problems today. The computer is very, very slow. It asks you a bunch of odd questions, and that will get at all the weird stuff we're going to talk about today. All right, this is, so I have you 3 hours today, so I'm just going to go at my own pace, and it'll be about 3 hours. The first 2 hours is epigenetics, and it is by far my favorite lecture that I give you guys because there's a ton of hype around it, but then there's a ton of potential. And so what do I talk about? As you'll see, I could talk about anything. So much is covered in epigenetics. So what I'm going to do is I pick a few high-yield pathways, and I'll go through those so you're learning those and they're board testable anyway. And then I'll talk through a few diseases that are high-yield, and then I may talk about some non-high yield stuff that's particular to epigenetics to let you understand it. And it's an amazingly fruitful research field, meaning it will change in the next decade and 2 decades. You're going to be having drugs based on this. So what are all these pictures about, aside from my delightful son? A ton of the information we have is in mouse studies. I'll talk to you about human studies too. Some people have said, so again, there's a lot of hype, so I'll mention the kind of anthropomorphization that people have had for epigenetics, but some people have said this is kind of Lamarckian winning, and he was the person that said giraffes have tall necks because they reach high for the leaves, which is incorrect, right? They didn't evolve because of that. But epigenetics does respond to our environment, and we can pass on to future generations things that happen to us in our environment. It doesn't go very many generations in the way most people talk about, and that's one of the key things to take, and I'll talk about the time frame of how long epigenetics can last. Some can last days. We'll talk about the circadian rhythm. Some can last a lifetime. Well, I think I took that question out. We'll pause on that. Very kind of high-level things that don't often relate to animal models affect our epigenetics. These horrible events such as 9/11 or war have epigenetic effects that we can follow. Famine. I'll talk about all those. And in a more kind of day-to-day life for all of us, our diet amazingly affects our epigenetics. So coffee, alcohol, fruits and vegetables, fats. I won't get into this really in this talk, but there's a whole science behind when you take medicine based on your circadian rhythm. And so there are certain drugs that are metabolized basically 50% if you take them at the wrong time of day. Why don't I go into that? Because it's different per drug, and basically you're going to follow the label. They should have done that, and if it says take it in the morning, prescribe, take it in the morning. But know that if they're taking it off cycle, if they're taking it some other time, maybe they're getting half a dose. That's about the largest effect I've seen. All right. Good, a bunch of people took it. And this should stay open for about 20 minutes, so if you want to keep going, it should be good. All right, so some of these objectives, as I mentioned before, I've given to you, so hopefully you already know some of them in the transcription translation lectures. So HATs and HDACs, what do they do? We're going to talk about Fragile X. We're going to talk about CPG islands. We're going to talk about modifying the epigenome, so how long does it last? There will be one slide about the cost of doing an epigenome. I've talked to you about doing sequencing. What's it like for an epigenome? Angelman and Prader-Willi is probably one of the, it's definitely board testable. It's tricky. And so when we get to that slide, I would urge you to get in groups again later and go over it. I have to sit through and work through it every time. So it's a great example, and if you get that, you can understand one aspect of epigenetics I'm going to talk to you about. We'll talk about some famines. I think I still have a slide about this. If not, I'll take it out. Epigenetic drug. I'll spend a bit of time on the circadian rhythm just to put this in a little bit of perspective. This lecture, first of all, it's 3 hours. We're going to go at whatever pace we want. So this is honestly a fun lecture. Stop me and ask questions now. A lot of you guys have a lot of the same questions. I think students interact with this material more because you don't have to be in a disease state to have this. This is kind of common stuff and it's fun to talk about. But back to kind of how it's a little different than your other lectures. This lecture used to be split. We had kind of the intro version in foundations, and then we had this more advanced version at the very end of year 2. And so we've combined it all, but you will get some kind of behavioral aspect that you could have seen later in your psych section. So we'll have some kind of clinical aspects like that. Wnt signaling is an example of one of those molecular pathways that I'll get into. I'll talk about alcohol at a very high level. You'll see these, you've heard me talk about sirtuins. You'll hear a lot more about that today in the circadian cycle. And the rest I'll leave for you to read. All right. So we're going to spend a lot of time talking about, so the epigenetics is a word that means modifying your DNA without changing an A, T, G, or C to something else. You might methylate it, for example, the cytosine, but you're not actually changing an A, T, G, or C by swapping. It's not a mutation, right? But there's different types of, so that's one type of epigenetics. Another type of epigenetic change is a histone modification. So it's all things that affect how much protein you might end up making or how much RNA you might end up making, but there's different flavors, right? So we'll spend a bit of time on this cytosine methylation because really it's by far the most well understood and it's very clear. It's easy to study, it's easy to board test, and I just think of it as, I think I have a poll everywhere, I'll give some of this away now, but I think of it as in the promoter region, so gene, promoter region, if you methylate the cytosines, think of them as bumps on the DNA, and so your landing gear of transcription factors can't land and you're shutting off the gene. So you're not going to make the RNA. So it's very simple, shutting off. We talked about the chromatin unraveling and modifying the histones, and you will in particular see these HATs and HDACs. HATs, histone acetyltransferases, turning it on, and HDACs, histone deacetylases are turning it off. And again, remember I talked through that should make sense because if you are taking off this acetyl group, you're making this lysine charged, and the positive charge wraps the negative DNA. Okay. All of that is review. I don't know if I mentioned long non-coding RNAs to you before. They are, I would say, not board testable because they are so complex. Dr. Gardner, have you ever heard of a board question on long non-coding RNAs? Yeah. So the long and short of it is there are a ton of them. So it's not that they're not important, okay? There are potentially more long non-coding RNAs than we have genes, okay, to put that into perspective. And they can turn genes on and off, and they do that in different ways. So, and they don't just turn genes on and off, they can interact with other things, not the direct DNA. So they're complex, very important, but we're just not there to understand them well enough or to test on them. Okay, so that's mostly review. Any questions? All right. This lecture was very different last year. It was a Prezi, so if something looks odd, ask or send me an email and I probably didn't catch everything. All right, so 2 ways that non-coding RNA can stop production. Sorry, let me back up again. The reason, I think I said this, there are drugs already out on the market that are, for example, histone deacetylase inhibitors. So you have to be able to kind of think, what is that doing? And they'll talk about, if they're using that for a cancer or whatever. Okay, so RNAi is interference. So you're interfering with making that RNAi. And it can go through this RISC complex that blocks or destroys the mRNA, or you can methylate the promoter and turn off the gene, okay? So methylate histones and block transcription, okay? Here's a recent paper, 2015, that shows siRNAs and miRNAs that are already as therapeutics, and you get a rough idea of where they are, okay? So cancer is over a quarter, I was surprised to see that ophthalmology has a lot of them, and then genetic disorders. So that'll change over time, and not board testable, but just so you know, they're out there and they're in diverse things. So if you come in and basically block that promoter from making the mRNA to block the protein, I think this shows exactly the same thing as the other slide. I'm just showing it. Okay, so this was, sorry, let me slow down. I have them labeled 1 and 2. I don't think this exactly follows my notes. I have all the same information in the notes, but not exactly the same. So showing number 1 here is this, if you want to visually see what it is. And then number 2 is shown here, okay? You get the take home that you're basically shutting it off. I think I've shown you this graph before, showing that as you increase in complexity, a higher percentage of your DNA becomes non-coding. And so humans have, it looks like 100%, but largely non-coding RNA. And that's a lot because of the long non-coding RNAs. Yeah, so the microRNAs can block long non-coding RNAs as well, so it can get complex. But the whole PowerPoint you guys have, if you guys want this figure, let me know. You don't need to know it, hence the big FYI. And the PowerPoint I gave you, I compressed so you could load easier. And then when I looked at it today, this figure is horrible for you guys. You can't really see what's going on. You don't need to know, but I just wanted you to get the point that long non-coding RNAs are all over the place. Again, if you want the figure or this paper, I'll put it on Canvas for you guys. Okay, so this should be yay. We don't need to know it. It goes up and down, and I'm not going to test you on that. All right, so that's largely review. Turn, talk to each other. I only gave you like 30 seconds for this. See if you can get that right. Ten seconds. Okay, so if that seemed like all review, hopefully that's good. If not, you can go back and look at the small interfering RNAs. Okay, and here's a cheat sheet. I don't know if I showed you this before, but you can just rote memorize this. The histone phosphorylation can activate histone acetylation and inactivate DNA promoter methylation, but basically you can see what protein's responsible and does it turn on and off the genes, okay? And if you're, stressed for time and boards, it's really hard to test long non-coding RNAs. If someone actually mentioned histone methylation, it's mixed, but it's more often off, and then the other ones are pretty clear, okay? So, even though this is foundations, you should be able to read a clinical question that mentions one of these in any random pathway you've heard or disease state and understand, okay, this is a HDAC inhibitor, so it's trying to activate a gene, okay? Okay, and all I want you to get, okay. The order of talks flipped around a little bit. At the end of today, I'm going to give you a molecular diagnostics talk, and I'll go through the cost of DNA sequencing in a little bit more detail. All I want you to get from this is that the cost of epigenome sequencing is similar to DNA sequencing in that you can get less detail for cheaper. So you can have predefined kits that do kind of a subset of epigenomes and it's cheaper, or you can do the top version, which is very extensive, but costs $5,000. And I think this is pretty recent, it's within the last year, I think. And the take home here is, unlike genetic sequencing, this is a bit away, meaning, let's say 10 years. But in 5 to 10 years, this could drop and it could be an annual visit. This never ends, right? You're sequenced once for your body, unless you have cancer. But this could be, okay, you have type 2 diabetes, we want to help you lose weight, here are your checkups, and we're doing epigenome sequencing of you at each check-in. Right, $300 is not that bad. Okay, so I think I used to have a poll everywhere here, but I don't think I do. So I'm going to ask you for a show of hands. What is the largest time frame that you think epigenetics can control? Okay, is it, and I'm going to go from left to right, so is it an evolutionary time frame, 3 generations, down to a day? Okay, raise your hand at the largest, okay? Evolutionary time frame. Okay, 3 generations, greater than 3 generations. Okay, about 5 and 5. 2 to 3 generations, about half. A lifetime, 5%, okay, years and day. Okay, so you had a bell curve around 2 to 3 generations, which is great. The right answer is you can control epigenetics on an evolutionary time frame if you think of X inactivation. We are inactivating things, and we have to do that every generation, right? So we have molecular machinery to do that. So there is epigenetics at every one of these levels. Most of the time, people are, most of what we really see is in a lifetime, and in humans, we see 2 to 3 generations, and there has been very little, but a bit of data on greater than 3 generations, and I'll talk through why that is. And, well, we'll get into that. And this worked better as a poll everywhere. Now that I look down and I tell you that I see that I gave you the answers. So here are examples of all of those, and we're going to work through them. Okay, so this is, I'll be very clear, this whole slide is not board testable, but I think is super cool. So it turns out that about 500 million years ago, there's something called whole genome duplication, where the whole genome can be duplicated, and normally the embryo is just gone, dead in the water. But once in a while, the whole genome is duplicated, and then one of them is completely epigenetically shut off. And so that organism lives. And then over time, that extra genome can be turned on, and things can survive. So it turns out that happened to us about 500 million years ago, twice. And so before that, we had something like myoglobin, and then our whole genome duplicated and we had 2 myoglobins, and then our whole genome duplicated and we had 4 myoglobins. And so those ended up becoming hemoglobin, myoglobin, something like myoglobin, and then we lost one. So, okay, so that's cool. So what? So here, they're looking at, on the x-axis, young versus old genes on an evolutionary time frame, okay, and young versus old gene. And here they're looking at methylation of the promoter region in A and of the gene region in B. And you can see that the methylation of the promoter region is conserved on an evolutionary time frame in the promoter region, and it's not in the gene region. And so this is that turning off of the genes was conserved on an evolutionary time frame, okay? That's just showing the same thing. All right, here is very board testable again. So there is a completely biological difference between men and women as it relates to epigenetics. And the long and short of it is that women affect 1 more generation. So that is Mr. Dr. Arbor saying that women have a bigger effect, okay? So if a couple is conceiving, and let's say they're doing something horrible. Let's say they're smoking or drinking heavily. If they're doing that a month before they conceive, and then they conceive, and the woman's doing it during pregnancy, well, let's take 2 cases. Let's take the first case where the husband doesn't do it. He's pristine, just, eats kale and water, and is great. And then the woman's a heavy smoker, okay? She is affecting herself. She's affecting her own genome. She's affecting her egg that becomes her baby. And then she's affecting the egg inside her baby that becomes her grandchild. So the moment that she, heavily drinks and heavily smokes, she's affecting 3 generations. But I want to make a point. This is why 2 to 3 generations is known. This is a single time that's affecting cells in the organism. It's a very different thing to get to that 4th generation, okay? Whereas a man that is, and there is data now that's come out in the last 2 years, that men, shortly before conceiving, what they do epigenetically affects their child. And so the data just came out that I've seen 2 years ago, but I kind of thought that was the case before I had my kids, so I was like super healthy. And then after my last kid, I was like, I'm done. And so I've been a little less healthy. But so there's science behind that, and it is better for both people to be in shape before having a kid. Okay. Any questions about that? Okay. This is a new slide I added, and this came out within the month, I think. So this is definitely out there. But it was just found that, so C. elegans is this model system, and this is different because it's a model system. C. elegans lifespan's about 3 days, so it's very different from us. And they even affected the promoter, I think. But they found epigenetic signatures that lasted 14 generations. So if you go back 14 generations, you're, like pre-colonial times. So the idea that when I first gave this talk, we talked about various cultural things that could play out here. So if anything close to that would apply to humans, then slavery would still be having effect, right? Clearly, the first and third world is still having an effect to this day. But so there's a lot of model systems where some of the science might have to be looked at in new ways. So for example, in C. elegans, scientists often need to go home and they'll put the C. elegans at a cooler temperature to like table the experiment. And now we know they're changing the whole epigenome of the C. elegans. So this was done, by, give me a sec. This was done by putting a red color related to a heat gene in the C. elegans. And basically you saw that the red color lasted up to 14 generations in the room temperature versus cold C. elegans. Okay. Any questions? All right, so here's another doozy to work through. So, okay, so we all have epigenetic effects, right? We have our genome, and then over time, our epigenome changes because we exercise or smoke or whatever. When an egg is fertilized, the epigenome is, the male epigenome is attacked and basically erased. And so that is this blue line. So the epigenome is erased from the male chromosomes. The female genome is epigenetically, over time, it kind of passively gets erased because it's not maintained, okay? So the timeline's a little different, but they both get erased. Now I'm gonna point out that this is above the x-axis, okay? So know that not everything is erased. That's gonna come into play later, okay? So why is it erased? Well, you're using epigenetics for the embryo to develop, okay? So your a lot of what you did, let's say if you were a smoker, hopefully can get erased, and then it's reprogrammed for the embryo to develop, okay? So this is post-fertilization reprogramming. Then, later on, there's a different reprogramming that happens to the germ line, okay? So if you have a daughter, her eggs have a different reprogramming. And so the point there is, and I'm going to show this in the next slide, that there's an imprinting if you're a male or a female. So we'll talk about parts of the chromosome where if you inherited that from your father or mother, one of them shut off and one of them's not. and it's dependent on if that chromosome came from your mom and dad. There's set regions, and we know those regions, okay? I doubt you'll hear this, but if you hear the term fifth base, the cytosine gets methylated when it's off, and then it can get hydroxylated, and that's kind of a signal that it's about to be erased, okay? The methylation's about to be taken off. Okay, so we talked about on an evolutionary time frame, exon activation. I think Dr. Gardner probably already talked to you about this, but exon activation expresses differently in different cells. And so here, I just found these patterns interesting. So cornea, skin, cartilage, inner ear, you see that the separate X's got inactivated, and then they kind of replicated, and so they created that pattern based on how they replicated. Okay, so we're still in the 2 to 3 generation range. And so you're going to see these regions here that have blue and gray are parental and maternal imprinting. So those are shut off in a parent or in the mother's chromosome or father's chromosome. And very unique sections are where they overlap, okay? And we'll talk about those. Why do I have these pictures? I know if you haven't heard of it, bees, when bees need a queen bee, the queen bee is created epigenetically. Likewise, you can have cat coloring due to epigenetics, and you have these different splits. So it's across the animal kingdom we have these. Okay, still in 2 to 3 generations or a lifetime. Well, this is more of a lifetime. There's been a lot of cool twin studies. So you don't need to know really what's going on here, but to see that on the right is later on in time, and red is different. And so if you compare twins and one of them's a smoker and one of them's not, you can see a massive epigenetic change. Or, it's not just smoking, but those are some of the best studies I've seen. So here's, again, the cytosine methylation. 3-year-old twins, very similar. 50-year-old twins differed a lot. Any questions? Okay, how's the speed? Fast, slow, good. Good? Okay. So one of the harder things when talking about epigenetics is that, and I am not faulting you at all. If I were a student, I'd want it too. Stuff's complicated and you want good, bad, right? Like, is this good or is this bad? And it depends on the situation you're talking about. So unfortunately, people think, okay, people evolved to be perfect, so if you're turning off a gene, that's bad. But that's not the case, because a lot of times we're not in our pristine condition, we're not in perfect metabolic health, and so turning off a gene may be good to prevent obesity, right? It gets a lot easier if you talk about cancer, because then you're saying a class of things is bad. So if you turn off a tumor suppressor, that's bad. If you turn off a proto-oncogene, that's good, okay? So that's one of the clearer kind of case scenarios to talk about. And there are drugs out there for it. Okay, so this is simplified as much as I could do. So there's a really cool study done called the Overcolic study, and in short, there was a famine, and they tracked generations, okay? And they found that if the grandparents underwent the famine, their grandchildren had cardioprotection, okay? They were protected from diabetes. And the anthropomorphic way of remembering this is it's as if our body is in a stressful situation, we epigenetically adapt to be in that situation again. Okay, so surprisingly, starvation can have beneficial side effects. And so I've already preached, you'll get this more in year 2 for me, but intermittent fasting has been shown to be effective, and you're kind of trying to toggle some of these epigenetic effects that you get from intermittent starvation. Likewise, a single parent, for some reason, I think the data was only statistical in the father. If the father was in the famine, his children were epigenetically protected too. Interestingly, if the parents were not starved, if they had food abundance, then their grandchildren were 4 times more likely to be diabetic. Okay, so I try to kind of push you guys to be, you're going to be the people that help set public policy. This needs to be thought of, right? If you are going to tax high-fat food, is it someone's fault if their grandparent was obese and they're 4 times more likely to be obese? It's in no way their fault, right? It's a societal thing that we have to change over generations. And then you get into like, food desert islands and poorer communities have access to worse, cheaper food, and so you're already 2 generations in the hole, okay? All right, so I wanted to show you the BMI chart since I just said obese, and I wanted to say I hate this. I hate BMI because it's just an awful tool, but it's easily usable, so we'll continue to use it. The reason we use this is because when people get checkups, they're weighed, and we know their height. But an Olympic weightlifter looks obese, and they're pretty fit. So here's an example of better things to do. So there's a body shape index. I'm a big fan of percent fat. These other things are easy enough to do now. If you're in a clinic, I would hope you invest in a percent fat machine and make that part of their electronic record. Okay, Dutch famine. If the mother, we're looking at different trimesters here of a mother, again, during another period of lack of resources, okay? If the mother was in the first trimester, her child was okay? If she was in her second or third trimester and she was during this Dutch famine, then this child was small for gestational age. And that, to me, makes sense, right? I think both my wife and I refer to the fetus as a parasite. The parasite's growing a lot in trimester 2 and 3, so you need a lot of resources, and if you don't have it, the baby's going to be small for gestational age. So if you look a generation further, interestingly, and I can't explain this, when the mother was in her first trimester and did not have access to enough food, the grandchild was small for gestational age. Okay, so I don't know. I don't believe there was I should look that up. I don't think they specified if that was for for both sexes. So, for example, if she had a boy, would that be the case, right? So that I don't know. I'll look into that. And then the second and third trimester was okay for the grandchildren. And then later on, they found out that the babies that were in that first trimester, the child of that, they did have an effect 6 decades later, even though they were not small for gestational age. So this kind of is less clear, right? Because I just kind of showed you something saying, hey, you can have cardiovascular protection, and if your grandparents had famine, and they can be obese if the grandparent was obese, and here we're showing that they're small for gestational age if they don't have that. So those are conflicting. I would argue the latter is more, the first one's more important. Yeah. Oh, sorry, it was squeaking. Let me turn it on. Go ahead.
Well, I was just curious. I mean, I imagine, did the famine only last a couple months? Like, how could you only be in one trimester during the famine?
It was a short famine. It was. This was the potato famine, I believe. Yeah. Was this, Dr. Gardner, do you remember the time frame of this? I thought it was like 3 or 4 months. Yeah, I remember it being a short period. I'll look up that time frame and put a Canvas announcement. It might have been, I forget if it was like at the end of a war and people came in with food or what. They've actually That was a different one with Russia, also had that, I believe. Okay, so here is probably one of the best slides, most confusing, but also if you get it, you get it. I think the only change I made in the PowerPoint that you don't have is this brown thing here, which is just explaining what I'm saying again, so you don't need it. So here are two syndromes, okay? I'm going to take at least 10 minutes to go over this one slide, okay? So Prader-Willi syndrome and Angelman syndrome. And what's happening here is this is one of those regions where the parental inheritance and the maternal inheritance overlap, but they overlap in opposite ways, okay? So here's this region, okay, and these, if I colored it green, that means that box is active, okay? It's not, it's normal. And if I colored it red, it's epigenetically shut off, okay? So from the father, the Prader-Willi region is active. So that means that gene is normal. If you have the normal gene, you do not have Prader-Willi syndrome, right? You have the syndrome if you don't have that gene, okay? If you inherit that same region from the mother, the Prader-Willi syndrome region is epigenetically shut off, and the Angelman syndrome is on, okay? So so you can inherit both of these. Take this part away. The issue comes in when one of them has a deletion. Let's assume there's no deletion here. Let's assume you inherit this and you inherit this from the mom. Then you would have active Prader-Willi from the father, and you would have active Angelman syndrome from the mom, and you'd have neither syndrome because you have both genes. When you have a deletion, it depends which chromosome is deleted. So if the father's chromosome is deleted, you lose the Prader-Willi genes that worked. And so you have Prader-Willi syndrome. This can happen in both males and females. Okay, and so I said the Prader-Willi goes with paternal, meaning the parent with the normal on gene, and if you delete that gene, you have the disease. On this side, we have, again, the same two, but we have a deletion from the mom. So you're deleting the Angelman syndrome gene that was active, and this person has Angelman syndrome, okay? In addition, in a rarer case, you can have uniparental disomy, and where basically you get two copies of the mom, and even if there's no deletion, if you had two of these, what would you have? Into the mic. If you had two from the mom and no deletions, would you be normal? Would you have Prader-Willi or Angelman?
Prader-Willi.
You would have Prader-Willi because you'd have two of these reds. You'd have no functional Prader-Willi. And you'd have two active Angelman syndrome. Okay? So turn, talk to each other, and then we'll go over it if need be. Turn, talk to each other. 10 seconds. That makes me want to proctor the exam to see if you do that. All right. And the answer, very good. Okay. Best class yet. All right. Well done. But I did say choose all possibilities. I'm going to have to reel that back in a little bit. Okay. So how could you have Prader-Willi? Do you guys want me to work through that? Yeah. Okay. All right. So, I said there's a male with Angelman syndrome. So here's a male with Angelman syndrome, okay? This is very rare, where it's coming from one parent. So this is what's going on, okay? If a male has Angelman syndrome, by far the most likely thing is that you have a deletion from the maternal chromosome, right, which had this positive region, okay? So the male, let's say the male passes on this chromosome, okay? It's going to be epigenetically the same because he's a male, okay? So he's having a child with a normal female. So the normal female does not have a deletion. So the normal female has a Angelman syndrome gene, right? What did you guys say? You said, right, so you got that right, that the child would not have Angelman syndrome. But if the father passes on this gene, this chromosome, the whole thing's deleted, okay? And then the mother passes on this without the deletion, so she passes on not epigenetically shut off Prader-Willi syndrome genes, and the father passes on a chromosome where that whole region's deleted. So you'd have basically this, Prader-Willi syndrome. Good? Good? No. No. Okay. Okay. So that pen thing said it wasn't working. I don't even want to mess with that, so I'm going to use my words. Okay. So is that everyone's good how you would not have Angelman syndrome, correct? Yes. Okay. And you're good how you could have neither. So, God dang it. Okay. So everyone's good that this could be a male that has Angelman syndrome, right? So he inherited this from the father, and he inherited this from the mother, right? Okay. So now, let's say he passes this chromosome on to his child. Do you see, this is deletion. This is deleted. So there's none of these. Now, what's the mother going to pass on? I want to... Okay. The mom's passing on this. She's passing on this not deleted because she's normal. Okay? So she has the Angelman syndrome, but she doesn't have the Prader-Willi genes. And the father has that. So there's no Prader-Willi genes. Functional. Good? Yeah. Sorry,
right,
they're still on. Yeah.
How can you have like maternal and paternal differences if it's not an X-linked? I'm a bit confused on how the mother passes on different active genes versus the father.
So do you remember, first of all, I didn't mean to do this, but for everyone, and I don't care how you learn, but for everyone that's watching at home, they didn't get to see my hand over the screen, just so they understand what was going on. Do you remember this? Okay? This epigenetic signature, you imprint, if it's a male or if it's a female, different regions are imprinted, which I showed, where did I show that? Sorry, this is going to suck on Panopto, me jumping around, but here. Different parts of the chromosome get epigenetically shut off if that chromosome is from the father or from the mother, even though it's not on the X chromosome. Okay? So think about how X inactivation happens. Now imagine, it's not quite that, but imagine that happening to certain parts of other chromosomes. And sometimes it happens here if it's from the father, and sometimes it happens here from the mother. So even though this isn't the X chromosome, so the reason this is such an interesting case is because, one, you have imprinting of the paternal and maternal overlapping one region, and you also have that region sometimes being deleted. I think this is a great slide to kind of run through multiple examples in a group, okay? All right, let's keep moving. I'm gonna go a little bit longer on this section and then shorter on the next. Fragile X, I feel like you've had a bit. So you have these CGG repeats. Once you get over 200, you have hypermethylation of those CPG dinucleotides, right? So there's a phosphodiester bond between C and G, and then the fMRI is shut off. FMR1. I don't know. I didn't show you this gel before, did I? No. Okay. So there are enzymes that can work or don't work on methylated DNA. And so here you can see a smear, and the smear is an affected individual that has more repeats, okay? So to be very clear, this is varying level of repeats going up, right? This EAG does not digest the methylated DNA, and so for the affected individual, you see this smear still there. I believe that's correct. I will come back and update you if I'm saying that wrong. So here you see again, there's an uptick because there's a repeat, but it's not, but it is being digested. So I don't think you would get a gel on the boards, but no, if you got it in a stem, you could get it in a stem, that there's some study done and they're applying a enzyme, and I would tell you anyway, that it does not digest methylated DNA, okay? If I'm talking about one row and I'm saying, look at this row and it doesn't digest methylated DNA, later today I'm gonna do the talk where I talk about gel electrophoresis. Okay, this is a slightly outdated slide, but I'll probably leave it in forever. This was a flyer that I was handed when Kathleen was pregnant with our first child. So we went to genetic counseling, and they handed us this, and they had Angelman syndrome, and they had Prader-Willi syndrome. They didn't have Fragile X. And I asked them some questions. First of all, our genetic counselor got the number of human chromosomes wrong, so that was depressing. So if you need to remember that, there's the company 23andMe, based on our chromosomes. Okay, so anyway, so I asked them, well, I couldn't believe that this test would test for any epigenetics. You could have mutations that are causing these, right, deletions. And so the long story short, the genetic counselor didn't know. So if you're in a clinic and you're providing flyers, know what the test is actually doing. So of course, being me, I called the actual company, which took a while, and a lot of people at the company didn't know what the test did. And eventually I got someone to say, no, we know we're not testing for epigenetics. We know that's a significant factor. And basically, if it comes up as maybe positive, we send them to a much more expensive test. And they told me basically they're selling something that's never used. It's all negative results, and this had never come up before. So anyway, know what you're selling your patients. And they interestingly were trying to upsell it because they didn't quite understand it, but they thought it was a quality of service bump. Okay, so now we're going to get to the kind of time frame that things are clearly happening in a lifetime or years, and where we start to have therapeutics, so cancer. So in breast cancer, these BRCA hypermutations or epigenetic effects are 13%, and colorectal cancer, there can be targeted to 40 to 90% of cases are these spontaneous ones. And so just going over, I think I have, so both of these have hypermethylation, okay? Both of these genes. And so a take-home, I think I have another slide, but in case I don't, I'll just mention here. A take-home is for cancer is that what you tend to have is you have hypermethylation of a very important gene that's protecting you, right? So you're shutting off that gene, and then you have hypomethylation of a ton of stuff. So it's temporally very important. You hypermethylate a protective gene, and then you hypomethylate proto-oncogenes and turn them on. Okay? So here's a bunch of cancers that have low global methylation, which is generally bad. You're turning on these things. Okay, so I do have it. So here's that drug, vorinostat. It was approved quite a while ago now, 2006, for T-cell lymphoma. It's an HDAC inhibitor, okay, so it increases gene expression. And it's 30% effective. So the take home I want you to get from this is that a lot of these epigenetic drugs they work, but they actually work better in tandem with something else. So vorinostat plus quality of care drugs had 85% remission rate, okay? So they're kind of orthogonal. I think I mentioned that word to you. They're kind of acting differently, and so they're synergistic. That's quite a few buzzwords I'm throwing at you, but they're very accurate. Okay. I'm going to go a little bit longer. Now we're kind of getting out of that to larger realms of how people do stuff, and this is going to start to become less board testable, okay? All these things up here that you have been told are good for you and they have epigenetic effects, okay? So green tea, soy, raspberries, broccoli, red wine, and a lot of these work by blocking DNA methyltransferases, okay? So we just talked about methylating DNA, they're blocking that. I will mention, if I don't have another slide on it, yes, that the amount you need of these good things to have the effect that we see in the lab is much more than anyone could consume. So for example, like there's been a ton of research done on red wine. Researchers love to drink red wine and they want to justify it, but to get the amount, so in red wine, there's resveratrol and lycopene, also in grapes. And so to get the amount that we're seeing in mice that's showing this positive effect, you'd have to drink hundreds of liters of red wine, okay, per day. So, so this is a dichotomy because we see these long-lasting effects like Mediterranean diet, right? We see things retrospectively where, okay, it seems like this population that has this, that is consuming this, has beneficial effects. And in mice, when we administer it, we see the molecular pathway up, and we can even see the molecular pathway up in people. But the dosing seems to be much more than is needed. So we are, I would say, still 5-10 years away, at least 10 years away probably, to getting dosing right on these types of things. And then there's also bioavailability. You probably won't hear about epigenetic bioavailability because it's so far out there. But for example, resveratrol isn't absorbed well. And so people have talked about how can you administer that. So they've talked about putting it in like chew tobacco because that's alkaline. And if, I won't call anyone out, but if you've ever done chew, it basically burns your mouth. And that's because you're breaking open vessels and it can go right into your bloodstream. So I would not personally take a little bit more resveratrol for chewing tobacco for the cost of that health-wise. But so long story short, I believe these things work on a molecular level. How to get enough of them and the right amount is still way out there. And a lot of times people are justifying what they want based on, you know, a horrible website that they've read that has science behind it, but like 1% science. Has anyone heard of Natural Medicine, the database and website? Yes. Where did you hear about it?
I studied nutrition.
Okay, so thank you. You're the first student in all these years that has heard of it. Marion, since day one, has paid extra money to have access to this database for you. So, so what this is natural compounds don't have to undergo clinical trials because they're natural, right? Like if it's a shrub, a person can eat it. They don't need to do anything. You can patent it if for a certain purpose. But so long story short, how do you know how good natural compounds are? Well, this used to be called the Natural Standard. Now it's called Natural Medicines. It's the best resource I've found. They kind of are like a Cochrane database for natural compounds. So I'm going to just click through a few slides to show you what you would get. If you look up something like chia seeds, it will give you the background, the safety, the effectiveness, dosing, adverse effects, toxicology, interactions, mechanism of action, which is interesting to look at, and then an evidence table, which is great. So you jump to the evidence table, and it has a whole bunch of studies. What's the N of the study? So this is pathetic, 27. Is it statistically significant? Yes. And then it has a general quality, and this is in the middle. So this is an example of something that doesn't have a lot of data behind it. If you look up grapes, like I said, resveratrol, there's a ton of studies, and it scrolls down and down and down, and the studies are much better. So 71, a large benefit, high quality. And so if you have patients, I don't know if you'll have access to this in the future. Actually, that's a great question. I'll try to ask that if you maintain this after you leave. But if you have a patient that's like, hey, Dr. Oz says blah, 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 which I'm not a fan of Dr. Oz, but you can at least give them data, right? Okay, this is what I know. And this is all an FYI. Okay, let's go 5 more minutes maybe. So about a year ago now, oh no, God, I'm old. 2 years ago, the first thing that was similar to the human genome was done for epigenetics. They did 111 reference human epigenomes. And here's the link, if you want to go to it, you can click per organ and find out what the normal on-off is for genes in that organ. And so I don't think you need this yet because people aren't having epigenetics done on annual visits, but they will as your doctors, and here's where you could compare it to be like, okay, is this normal? There's a lecture that you will not have, I don't think. We've tried to have it a few years in a row, but we haven't gotten the speaker here. It just hasn't worked out. And the long story short is our interaction with these bad things in the environment is very complex. And so you should try to limit these interactions with bad things, but they overlap a lot. So here are things that affect your epigenetics. So here are 198 plastics that affect things, but you see they overlap with hydrocarbons and pesticides. And so if you take away a lot of plastics, you still have these 3 that overlap from hydrocarbons, from oils or whatever. And a lot of this is found near farmers or, like where they're very closely exposed. But that's honestly just because they're exposed to such high concentrations. We all kind of have this soup mixing up our DNA at some level, okay? All right. Okay, great. So now we get to see the results of what you guys thought. So a lot of this stuff I'm going to talk about in the next half. So we'll take a break. I just want to show you the results before the break. So in the next year, how many of you are going to say more positive things than negative? That's good. I'm going to talk about actually how words can affect your genome. Drinking more than 1 cup of coffee. Coffee's good. Sleep less than 7 hours a day. That is not good. I expected that bar to be high. I'll try to show you the data around how you are increasing your cancer risk. So the daily recommended vegetable intake is now 8 to 10 servings. FYI, I don't think many of us hit that, okay? People are still voting? Really? Okay, the ideal weight you'd like to be. let's see where we are here. I expected us to be around this, 5 to 10. Okay, so now the reason I wanted to show this anonymously was that I would say few are at their ideal weight. So it's not enough to, it's hard. It's hard in either direction if you want to put on muscle or lose weight. And so you're going to have the students we've had when they go in the clinic, they come back and just the number one thing I hear is, it is amazing how much of what I see is just lifestyle, type 2 diabetes, right? It's all these things. And I'm a fan of the ACA. You guys can work to affect public policy so as a city, there's incentives to increase the health quality of the city, right? That's very hard to do on a policy scale, but people have tried to start doing that. So some insurance companies now will pay to send patients home with vegetables, and they'll cover that, and then you have to track. They don't do that here. I don't believe in Indiana. In New York, they do that. But then you'd have to track probably the percent fat, cholesterol levels, whatever. Okay. All right, what should be taxed the most? Alcohol, number one. So you haven't read the red wine literature, obviously. No, so you're right that alcohol is generally bad, but 1 drink a day doesn't seem to be too bad. Soda, very bad. Sugar, horrible. Herbicides and pesticides, I agree, very bad. Diet soda, I would argue should be lower, although I was talking to Dr. Cumbe back there recently that a recent study came out showing that people that drink diet soda don't actually lose weight compared to people that drink soda. In fact, sometimes they can gain weight, which is very interesting. And no one wants to tax ice cream, shift work, salt, or cars. That's very interesting. I'm going to talk to you about shift work. All right, and which should be subsidized? Behavioral therapy, excellent, that's heartwarming. Okay, I'll talk to you about that. Physical therapy, I thought might be number one. It's osteopathic. Fruits and vegetables, clearly. Bicycles, interesting. HDAC's right. That's kind of the free market, but that's probably out of all of these, behavioral therapy, physical therapy, and maybe these drugs would be the ones you're most to. And I'll talk about gastric bypass surgery. It is amazing. It's like just one of the best things, and I'll show you data on that. And everything should be subsidized. Okay, one more. Okay, what did I ask here? What laws should be allowed? Okay, should be allowed or not allowed? Physical abuse of 2-year-olds on top? I think you didn't understand this, or I wrote it wrong. We're going to have to skip over that one because I'm assuming that was not your right answer. Okay, but we'll talk about the behavioral aspect. Okay, that's it. Take a break. I'll play some funny videos for you during the break.